Good morning, everyone, and welcome to day 3 of the 2021 Virtual Crop Diagnostic School. My name is Mike Brown. I'm a crops extension specialist with Saskatchewan Agriculture in Humboldt, and I will be chairing today's event. This is year 2 of our virtual format of our normally in-person annual event that will allow us to share diagnostic information from the field while ensuring that everyone remains safe. The Crops Extension Specialist team with Saskatchewan Agriculture are proud to partner with the Irrigation Crop Diversification Center, ICDC, based in Outlook, Saskatchewan, who are kind enough to seed plots and allow us to film various segments, which you will see today. ICDC is part of the AgriArm network, 8 producer-directed applied research and demonstration organizations located across the province. For more information on ICDC and the AgriArm network, please visit irrigationsaskatchewan.com and agriarm.ca. There's also a video in the YouTube playlist for an overview of the ICDC station. We would also like to thank our sponsors of this year's event, Sask Pulse Growers, Sask Canola, Western Grains Research Foundation, Sask Flax, Sask Barley, Sask Wheat, Prairie Certified Crop Advisors, and Saskatchewan Crop Insurance Corporation. Today's session will be on herbicide resistance and herbicide injury. We'll begin the day with an introduction on herbicide resistance, as well as a short pre-recorded video on how herbicide resistance testing is done at the Provincial Crop Protection Lab in Regina. Following this, we'll have live webinars on herbicide-resistant research and herbicide injury diagnostics. Each presentation today will be followed by live Q&As. The main hub for today will be your webinar screen, and we'll share relevant video links with you there. Today's presentation will be recorded as well. If you want to rewatch the recording, the video links will be sent out later this week, along with event evaluations. Filling out these evaluations helps us determine what topics you want to learn more about and helps prioritize research needs for the future. So if you could fill them out, that would be awesome. A reminder, if you had entered your CCA number during registration, we're going to send those along for you to receive credit. Credits are only available to those watching the webinar live. There are also downloadable handouts available in the handout section of the control panel, including an agenda and information on your local crops extension specialist. A few housekeeping notes to keep in mind during the webinar. Refreshments can be found in the handout section of the webinar with a fine selection of PDFs to choose from. Washrooms may or may not be located nearby, depending on your location, and the emergency exit is the X in the corner of your screen. If you have any questions during the presentations, you can type them into the question box in the bottom right-hand side of the control panel. We'll try to get to as many as possible, but may need to limit them due to time. Questions will be anonymous, and during the presentation, you will also be muted to limit any feedback. So with that, we will begin with an introduction to herbicide resistance with Shannon Chan, Crops Extension Specialist in Swift Current with Saskatchewan Agriculture. Before working for the provincial government in 2006, she completed a Master of Science degree in Plant Sciences and grew up on a grain farm southwest of Regina. Shannon, we'll let you take it away.
Well, good morning, and thank you very much for the introduction, Mike. So as Mike said, I'm going to talk about an introduction to herbicide resistance. So what's the difference between herbicide tolerance and herbicide resistance? So herbicide tolerance is the inherent ability of a species to survive and reproduce after herbicide treatment. So it normally tolerates a herbicide. An example of this is broadleaf weeds normally tolerate a Group 1 application of a product. Herbicide resistance is the ability of a plant to survive and reproduce following exposure to a dose of herbicide, normally deadly to the wild type. And the wild type is just kind of what the original type is. So in this case, it would be the susceptible weed. So the herbicide once worked on this, and now it doesn't. And I'd also like to say thank you very much to Clark Brenzel for most of the content in this presentation. So what does a herbicide need to do to kill a plant? It has to enter the plant through the leaves or the roots, move to the target site for that specific herbicide, and then bind to the target site and be active. So in a normally functioning enzyme system, you'll have building blocks coming in and then going back out, and that will produce a product, usually an amino acid or a lipid that's necessary for the plant to survive. When you have a herbicide, so that's the red here, it's binding, it fits into that specific enzyme system and locks in. So it doesn't allow that essential product to be built. And in addition, in some enzyme systems that herbicides will attack, one or both of the building blocks can build up when the herbicide is blocking the enzyme, and they can be toxic to the plant. And some examples of this are group 5, 6, 7, 10, 14, and 22. And most often, this is expressed as a loss of cell membrane structure, which means you have a fairly quick dry down. So I'll go into a few mechanisms for herbicide resistance, and these are just mechanisms that I found. It doesn't necessarily mean that we have these in Saskatchewan. We do definitely have some of them. So an altered target site. So in the resistant weed, some part of that enzyme that the herbicide attacks becomes altered. So the herbicide can't bind and the building blocks continue to come in and produce that essential amino acid or lipid or compound. And eventually, the herbicide will be metabolized by the weed. You could have numerous copies of the target site. So in this case, the herbicide goes in, still binds, but the plant has enough copies that even when all the herbicide's bound, it still can create the building block, or sorry, it still can create the products that are needed for the plant to survive. An example of this is glyphosate-resistant kochia, and it has 3 to 10 extra copies of the EPSPS enzyme system. You can also have enhanced metabolism, so the plant quickly breaks down the herbicide into different components that don't harm the plant, and this happens before the herbicide can get to the target site. We also have reduced translocation, so say this is a spring application of a product, it goes to the leaves. Normally, this would, as an example, this would be a herbicide that goes to kind of the growing points, and that just becomes stopped or slowed down. You can also have compartmentalization. So the herbicides are moved from the actively metabolizing parts of the cell to non-active parts of the cell. So that includes a vacuole possibly or a cell wall. There can also be sequestration. So the herbicide gets bound to another compound, sometimes a plant sugar compound, and then the herbicide is inactive. So how does resistance increase? So you have an application, maybe early on, you only have one plant that survives. So that plant grows. It'd be really nice if a cereal only had 4 seeds, but obviously there's a lot more than that. But those weeds survive. And then when you have the next herbicide application, you will end up having those 4 will survive and produce more seeds and kind of repeat. So how fast does resistance increase? This is as an example, if you have one plant in a million that has a mutation to make it resistant to the herbicide, for your first to third application, you'll have excellent to good weed control. You may notice when at the 4th application, there may be a resistant patch. But your 5th application, you have complete weed control failure. So 60% of the weeds are resistant to that herbicide. So to look at this more visually, here's the base population you wouldn't be able to see. So this is the same one plant in a million. First application, second application, there's a couple, 2 or 3 plants. Third application, you may notice a resistant patch, possibly, but often there's also other factors going on that may explain why the herbicide didn't work in that spot. And then this is the 5th application. So this is 50 of the half of the dots filled in, changing, going from green to yellow, and this is a weed control failure. And it's also possible sometimes you can get a spot where the resistant weeds are kind of concentrated. For an example, where a combine comes into the field and is turned on again. So how can you tell if there's a sprayer miss or if there's herbicide resistance in the field? So sprayer miss will have a geometric pattern in the field that has sharp defined edges, straight boundaries, parallel lines the width of the sprayer, and multiple species survive in that missed area. So just as an example, this is your field, and this is one where it got missed. So you have, just in this example, cleavers, flicksweed, kochia, stinkweed, and wild buckwheat. If you have herbicide resistance, it doesn't have sharp lines. So your boundaries are less defined. It may show a general pattern following the combine or other disturbances. And if you suspect that some weeds of one species are resistant, herbicide injury that causes plant death should be present on other plant species that are nearby in the field. So just as an example, in a field, maybe you have some flicksweed, some stinkweed, some cleavers, some kochia, and some wild buckwheat, kind of all spread out. The flicksweed gets controlled, the stinkweed gets controlled, the cleavers, the kochia. I just wanted to put somewhere where kochia was controlled. And even sometimes some of the cleavers would survive, but not all of them. You could actually have a live plant right beside a dead plant. And I'm sure everyone's seen the kind of the tumbleweed resistant patterns. This picture was from a few years ago. But as you can see here on the picture on the left, there's tumbleweeds that are actually going in two different directions following the wind. One of the things that for kochia that makes it a good spreader is it actually will dry down and break right above the soil surface and then just move with the wind. So if you see this in your field, this is something you should try to get rid of right away if you can mow it, even tillage if you have to. So you don't want to wait until the next year or the next herbicide application. So thank you very much, and I'll turn it back to Mike.
Awesome. Thanks for that presentation, Shannon. So before we go into Shannon's Q&A, we're actually going to watch a short pre-recorded video with Clark Brenzel and Scott Hartley, and then Clark and Shannon will be available after the video for questions. So Clark Brenzel is the Provincial Weed Control Specialist with Saskatchewan Agriculture. He is based in Regina and provides information and recommendations on weed control to producers, agronomists, government departments and agencies, as well as municipalities. He has experience in weed management with the crop protection industry, university research, and government. He has been with Saskatchewan Agriculture in this position since 1998. As part of this role, he also administers the Weed Control Act, raising awareness about invasive species and assisting municipalities and landowners in understanding their respective roles and responsibilities under the Act. Scott Hartley is the manager of the Ministry of Agriculture's Crop Protection Lab in Regina. He was also the insect and vertebrate pest specialist for the province from 1998 to 2017. Prior to this, Mr. Hartley was involved in insect research with AAFC in Saskatoon and other private contracts for projects including the Saskatchewan Wheat Midge Survey. Mr. Hartley received his formal education from the University of Saskatchewan. So you can go ahead and click the link provided in the control panel or in the email that we sent this morning and watch the video on your own device, as we may have issues with the video quality if we stream it through the webinar software. And after we've watched the video, we're going to return back to this webinar screen and we'll have a live Q&A session with Shannon and Clark. So we will see you in about 11 minutes here.
Hi, I'm Clark Brenzel. I'm the Weed Control Specialist with the Saskatchewan Ministry of Agriculture. We're here in this field of wheat today that has a few weed challenges. And one of the things we need to be aware of is that when we have escapes in our fields like this, we have to consider the idea that there could be resistance in the field. So we've got some wild oats here today that we'll look at and look at sampling methods for collecting samples to go to our crop protection lab for herbicide resistance testing. And we'll look at some other methods for other different weeds, and hopefully that'll result in you having a good sample going into our crop protection lab for testing. Okay, so we're in this field of wheat looking at our wild oats here, and what I like to use for wild oat collection is actually an insect sweep net. It's really handy because wild oats are really prone to shattering, and so if you've got grassy weeds that are very prone to shattering, this is a really good tool in trying to get those seeds out of the head of the plant and into the sample without a whole lot of other extra junk. In some cases, what happens is people just run their hands up up the top of the plant like that and pull the sample out, but they end up with a lot of chaff. And we don't want a whole lot of that chaff at the crop protection lab because it causes a lot of problems with us trying to get the sample prepared. So the sweep net is actually a very good way to conduct that sampling on wild oats. And the other tried and true method would be just to take the plants, especially if you've got plants that are tumbleweeds like kochia, just collect those when they're a little bit prior to maturity, and just like you would with canola sample, take those plants, cut them off with a pair of clippers, and then put them into one of these poly seed bags or an old pillowcase or something like that to allow them to dry down. And then once they're dry, you can do a bunch of stomping on that bag, and then that sample shatters out into the bag and you just get the pure sample of seed to take to the lab. A little more difficult to collect, say if we've got plants like Canada thistle here and some of our sow thistle in the foreground. They're wind-borne seeds, so a battery-powered cordless vacuum is actually a really good way to sample those plants and get the seeds off of the plants without a whole lot of extra debris and without losing a lot of the seeds that will, as soon as you touch them, want to break away and fly away from your sample. You can approach the plant with the vacuum on and then draw all those reed seeds into the vacuum cup, and then you've got a canister ready right here that's full of those weed seeds, and you can tell what you've got in the canister just by looking at it. Question of how much seed do we need for a sample? And for wild oats, what we're looking at is about a large coffee can volume of wild oats without chaff in it for our sampling. So we're looking at anywhere between 1,000 and 2,000 seeds, and that's roughly what will fit into our large coffee can. For smaller seeded weeds, so things like mustards and things like that, we're looking at maybe a smaller coffee can, so one that's a little bit shorter and narrower, and that gives us adequate seeds to be taking or doing that testing. And if we're looking at shipping these samples, we want to make sure that our samples are dry. We want to make sure that they're clean so that we don't get mold in transport. And the other thing that is one of our banes is the plastic Ziploc bag. One of the problems with plastic Ziploc bags is that they don't breathe. And if you've got samples that are the slightest bit wet, then what happens is that they get into the bag, they start molding, and you lose the viability of your seed. There's other ways that we can lose viability as well, is that these things, when they're all zipped up, they also make great greenhouses. So if you throw them on the dash of your pickup truck, you end up ramping up the temperature in that bag very dramatically, even if your windows are open, and you can end up killing your seed, and then you've got a sample that comes into our lab, looks really good, but the sample is dead as a doornail, or the ones that aren't dead are so dormant that we can't get them to germinate. So those are some of the things that are tips for getting samples to our crop protection lab in good condition and making sure that we can provide you with a really positive sample and positive results from your sample coming into the lab so that you know exactly what's going on in your field so you can make good management decisions.
Hello, I'm Scott Hartley. I'm the manager of the Crop Protection Lab for the Ministry of Agriculture. We're located in Regina on Park Street, 1610 Park Street, and we offer a number of services here in the crop protection industry. So one of the main things that we do over the winter months is herbicide resistance testing. Herbicide resistance testing is something that is important in farming today due to the buildup of resistant weeds to various groups of herbicides. So what we offer in the lab here is testing seed that's collected by producers to see if they truly do have resistance. Just a small amount of resistance showing in 1 year can really lead to problems in the future years. So this service gives them an idea of the level of resistance that we're noticing in the specific group of herbicides that they request. So what the lab requires for the herbicide resistance testing is, of course, the seeds. We do a number of different seeds. Primarily, the submissions have been wild oats, but we also do broadleaf weeds like stinkweed, wild mustard as well. We're hoping to get started into kochia testing as well this fall. So seeds have to be submitted to the lab. We request 2,000 seeds per subgroup requested, and this should go with a form, and the form has all the information on submitting the seeds as well. The form's available on saskatchewan.ca under just an easy search for Crop Protection Lab guidelines and forms for submitting samples. Provides all of this, as well as our other services besides herbicide resistance testing. So once the 2,000 seeds come in, we request dry, mature seeds for each subgroup. The reasons for this is that if it isn't clean, one, it's going to take extra threshing time and it can delay things in the lab. And the other thing is that if they're not threshed, it's hard to tell for sure if they are truly mature seeds. And if they're not mature, they won't germinate. So these are all things that are necessary in order to make sure that it's an effective test. So once seeds are submitted, and usually this is going to be during harvest time, so we're starting to see seeds August or September. They won't be processed, however, until January. The reason for this is usually because of the vernalization that's required for the seeds. If we seed them too early, we are not going to get sufficient germination to be able to do decent testing. Another reason for the 2,000 seeds per subgroup, just to be on the safe side. So in January is when we do start the process, and for most subgroups, we're actually seeding them in soilless mix, growing them in a growth room, and then spray with the appropriate chemical at the time that they're at a sufficient stage. There is another method we use for some of the subgroups that's an agar method, where the seeds are plated in an agar solution that's impregnated with the herbicide as well. So that's how the process goes. It does take time, and probably the biggest key is that it doesn't even start till January. So through this time, it takes a while for them to grow up to the appropriate size. It also takes time to do the rating. So rating is done based on dead plants versus surviving plants, and compared with susceptible and resistant control seeds of the same variety. So the reason we require forms, I guess primarily, is to be able to contact the producer with results when they're ready. But the other thing is there may be other reasons for it, like is there insufficient seed or poor germination, or more time required for the test. So the form provides that information for us to be able to contact the producer. And then once results are ready, our new system is that we contact the client, the person who is going to pay for the processing, and they contact our new accounts billing department. And then once the payment is made, the results are sent out to the client, and we're trying to do this in as timely fashion as possible. So a big change this year with our herbicide resistance testing, though, is the deadline for submission of samples. Where previously they were accepted later, now our deadline is January 15th, 2022 for this next growing season. So thanks very much for listening, and appreciate all the submissions we get, and keep in mind that there is other services besides herbicide resistance testing. So feel free to contact the lab through our email address, and also through our main lab phone number.
All right. welcome back, everybody. We'll get into the question and answer here with Shannon and Clark. Just a reminder, if you have any questions, you can enter them into the control panel and I will ask them on your behalf. First question here, I guess whoever can jump on this fastest can answer it, but the first question is, where can you purchase a sweep net?
Sweep nets are available. There's a company called Pro Metal, which is in Regina. If you search, I think it's PRO-Metal is where we've got, in the past when we've sold them, that's where we've got them from.
And that company also sells the replacement nets, because after a period of time, you will get some wear on those cloth netting and you'll have to replace them eventually.
Awesome. All right, let's go up here. What herbicide groups normally would not have a residual issue, but with the drought this year may have problems arising next year?
Do you want me to answer that one, Shannon?
I think that
Yes,
might go
please.
to Clark.
Yeah, I think that's a Clark question. Thank you.
Okay. Generally, we'll have some group 2s that typically under a wet year wouldn't have any concerns. So things like the imazamox-based herbicides, so things like Viper and Solo and all the generics that are sold beside those. Other things that may have residues are things like Curtail M may have excessive residues going into the next year. You may have things like Infinity have excess carryover into the following year after a dry year. You may find that things like Everest and Sierra also have flucarbazone-based herbicides may have more extended carryover potentially into the following year, and more a broader spectrum of sensitive crops, let's put it that way as well.
Awesome, thanks. Next question here, for the wild oat samples, other than a coffee tin, what would be an acceptable container to send the sample in?
Oh, I would suggest like a paper lunch bag or grocery bag. Lunch bags are a little on the thin side, so those thicker grocery bags are often very good containers. Any kind of, if you've got a short mailing tube, that would work. If you've got a box of some kind that's lined with paper, then that would work as well. The other thing that we find is that it's more the solid Ziploc bags that are a real challenge with breathability. But there are Ziploc bags that are specifically designed for storing vegetables. And those Ziploc bags actually breathe. So they've got little micro holes in them. So those are suitable for shipping as well. The other thing you have to keep in mind is that when you send things by courier, sometimes those bags get zipped up tight, airtight as well. So you may have to send it in a box so that they don't go into those sort of document shipping bags that get zipped up and are plastic as well. So just kind of keep that in mind.
Awesome, thanks. Well, that was it for questions. So we'll thank Shannon and Clark for their presentations. Next up, we're going to hear from a trio of Agriculture and Agri-Food Canada scientists who will speak on current herbicide resistance research. So I'll introduce all three and then we'll get in. Sean Sharp, he is a research scientist with Agriculture and Agri-Food Canada in Saskatoon. His program evaluates weed ecology and integrated weed management strategies to provide control of troublesome species and reduce the risk associated with herbicide resistance and environmental stressors. Charles Geddes is a weed ecology and cropping system research scientist with AAFC in Lethbridge. His work focuses on herbicide-resistant plants, weed ecology, biology, and integrated weed management, weed seed bank dynamics, crop weed interference, and long-term agro-ecosystem experiments. Brianne Tideman is a research scientist with AAFC in Lacombe. Her research focuses on integrated weed management, weed biology, harvest weed seed control, and managing herbicide-resistant weeds. She also conducts research on agronomic decisions for crop production and weed control. And we'll start off here with Sean and work our way through. Sean, whenever you're ready, take it away.
All right. Good morning. Is my screen coming through okay?
Yep, looks good.
All right, thank you. So today I'm going to start by presenting some results from the latest round of surveys for kochia. And these were initiated by Hugh Becky prior to his departure, so I sort of inherited these. There we go. So this is the map. So what we had done is we traveled out to different RMs throughout the province, and we collected 1 to 3 samples per RM, depending on how much of that RM is cropped. And then we brought them back to the lab and we screened them with a single dose of glyphosate. And the main takeaway here is that many of the populations are showing a low to high degree of resistance. And while there is a bit of grouping across the entire study area, we are finding the presence of the resistant biotype. So to compare to what was presented earlier, I would say we're at about, so if you recall the presentation slide that Shannon had presented, and we look at how many applications we are from losing full control, I would say we're at about 4 applications. So we're on the brink of something quite bad. So during the field collection, people had noted where the samples had come from, and unlike previous surveys, most of these samples had come from either in the field or in the ditch nearby, and there were cases of it being in both. So it's not a case of it's on oil sites or other non-cropped areas. Like these samples are coming, or they're in the field, unfortunately. So this was taken at about halfway through the screening. We're a bit farther now, but I'm anticipating to see that these results will stay consistent. So the second study that I want to talk about was inspired by this, and I wanted to exploit that with kochia, it doesn't tolerate being very low in the soil profile. So it doesn't tolerate being buried. So I wanted to apply some mulches on top of established kochia patches. So we had black plastic mulch, and I used a hydro mulch that was a coconut base, and we also used chaff that we had gathered either in the field or in the field margins, depending on how far along the field was into planting. And we also had a mowing treatment as well as a control where we overseeded with a perennial grass. And unfortunately, we didn't get good establishment as of yet with the perennial grass, but what I'm most excited about is that with the chaff, we were able to stunt and stop a lot of the weeds from emerging through it. So we are at over 8,000 plants per meter squared, which is very dense. And then with both the black plastic mulch and the chaff treatments, we were able to get them down considerably. And I just want to acknowledge all of the funders for these two projects, and I will pass it over to Brianne. Thank you.
Brianne, I think you're on mute still.
Thank you. I thought I hit the button. Apparently it didn't unmute. My apologies. Let's back up. So I'm going to give a quick overview here of the weed science research that's happening in Ag Canada Lacombe. So starting with integrated weed management, we do have a focus on both managing and mitigating herbicide-resistant weeds through the use of integrated weed management strategies. And this is really continuing work that was started by Dr. Neil Harker. And so looking at chemical, physical, cultural, and biological strategies that can be incorporated into our cropping systems to manage these weeds. So what does that look like? That looks like this. Using things like perennial forages, increased seeding rates of something like a spring barley or barley silage, as well as winter cereals. These are all strategies that Neil had incorporated into cropping systems for management strategies, and one of the things that we've added is looking at things like chaff collection or other harvest weed seed control methods, which I'll talk about in a minute, to also assist in management of those weeds. Now, one of the other differences that we've had from some of the work that Neil did is Neil tended to focus his studies on wild oat alone. And one of the steps we've tried to take, at least with some of our newer studies, is looking at sort of the community of weeds. So looking at some of those other problematic broadleaf weeds as well. So we've got everything from wild buckwheat, cleavers, hemp nettle, to shepherd's purse, volunteer canola, and Charles's and Sean's favorite of kochia. Only at the locations where we have kochia. We don't put that anywhere that we don't have it introduced yet. So we're trying to build on some of the work that Neil did, showing how these integrated management strategies could be used on wild oat and take them to the bigger wheat community, as well as adding any new strategies that we can find. I'm going to leave this study at that for now and come back to why I'm not talking a lot about the results as of yet. This study was a 5-year study that the field work was done in 2020, but we don't quite have all of our final answers yet, and I'll show you why in just a sec. So another big area of research for us is harvest weed seed control. If you're not familiar with harvest weed seed control, these are strategies from Australia. They've essentially all been developed in Australia that looks to manage seed bank returns. So rather than spreading those weed seeds that are left in the field at harvest time across the field by broadcasting them through the chaff spreader, trying to find some form of preventing that return, whether it's getting them off the field, whether it's destroying them, just something to stop those seed bank inputs. So we've been working on some stationary and controlled trials, and I'll show you a picture of those in a moment. We have also run a field scale trial with the tow behind Harrington seed destructor. So that's that big giant machine in the background there. That was the original tow behind seed destructor. These are not even available on the market anymore. Now the newer versions, the new prototypes are actually built right into the back of a combine, which is obviously much more efficient and a little more user friendly. But we have the tow behind version. They're equivalent in terms of control, so that's what we've been using. So we did run that machine in 20 producer fields around Lacombe in weedy patches, and we had comparisons to just a regular harvest where those seeds were spread instead of milled. And we did our data collection between 2017 and 2020. This is another one that I'll talk about, sort of why I'm not giving you final results quite yet. But we are also coordinating with researchers from Australia and the United States who are working in this area for what types of research we need to look at moving forward to try and sort of gain an understanding of how this could work in Western Canada and what weed seeds we can target and those types of things. So we do hope to have the results from these studies as soon as possible, but a big portion of the data from those studies comes from the actual seed banks. And so that involves doing seed bank grow outs, which is what you can see happening in our back room right here with all of our fancy grow lights, where we're actually going through cycles of growing these plants out, counting what emerges, they go through a freeze-thaw cycle, they come back in and get grown out again. So it is a very extensive process. We don't have a greenhouse in Lacombe right now, so we don't have the ability to do a whole ton of these at the same time. So we've got our little grow light setup that we're working with and slowly but surely working through all of that. But until I have that seed bank data, I can't really say how well I've done at driving down the seed banks. So hopefully you'll be hearing results from these very, very soon. We are working on it and counting and growing things as fast as we can. Now, we have done, as I mentioned, some stationary work with Harvest Weed Seed Control, looking at how well do these machines actually work on what goes through them. So I've personally done testing on the toe-behind seed destructor and the Redicop seed control unit. Just for those of you that may not be aware, Redicop is based in Saskatoon, so it is a Saskatchewan company. They do have a version of one of these mills. And basically what we saw with both of these units was that of the weed seeds that are going through the mills, you're going to basically destroy more than 95% of those. There are similar values globally that I've seen for the other brands of these integrated mills, the Seed Terminator, the integrated Harrington seed destructor, which as I mentioned is the one built into the combine, that updated version of what we have, the seed control unit, etc. So all of these mills seem to be doing a very good job. The Weed Hog is the newest brand of mill on the market, which I have heard is supposed to be slightly lower efficacy, closer to 80 to 85% control of what's going through it, but I haven't seen that data published as of yet. But basically what we're seeing with this equipment is if you can get the seeds into some of these mills, they will be destroyed, they will be killed. So the trick is just getting it into those mills to control them. Some other areas of research that we have as well, we do work quite a bit in integrated crop protection. So I work a lot with Dr. Hiroshi Kubota, who's the Sustainable Cropping System Scientist in Lacombe, Dr. Kelly Turkington, our plant pathologist, as well as various Ag Canada entomologists. And really we're asking questions on how do changes in an IPM practice change the rest of the cropping system. So as an example, in that big integrated weed management study that I was talking about, where we've got different rotations and different seeding rates and silaging and things like that, not only did we look at the weed management component, we've also done some soil microbial sampling, we've done some pathology sampling, looking at leaf diseases of the cereals and things like that. So we do try and incorporate the knowledge of some of those other disciplines into, if I make this change to control my weeds, what happens to all of these other pests that we deal with as well. Now we also do some work in weed biology and ecology, and obviously in collaboration with Sean and Charles, but I did want to talk about one new project that we're just starting, which is looking at cleavers and comparing various populations of cleavers to try and understand some of the diversity that we're facing in this species across the prairies. So we're going to be looking at things like emergence of the different populations, seed production, how many of these populations are prone to fall emergence, those types of things. We're going to do some preliminary screening with quinclorac for resistance, since there has previously been a quinclorac resistant biotype found in Alberta. And we're going to try and use this information that we're getting on how these populations vary to develop different management strategies. Now, the reason I bring this up is we are actually looking for populations from Alberta, Manitoba, and Saskatchewan. So if you are a Saskatchewan producer that has a cleavers problem, we would be happy for you to rip some of those out and stuff them in grocery bags and send them to us for use in this study. I figured it didn't hurt to throw this plug in there. So really what we're looking for is about 5 paper grocery bags of plant material with mature seeds on them from a field with a GPS location to go with it. If you want any more details, my email address is there on the screen, and you can get in touch, and we'd be happy to take some of your cleavers off your hands. With that, I'd like to thank the technical staff, the summer students, the co-op students that I work with. It's really through them that I have all these projects to talk about, as well as the many, many funders of the different research program or projects that I discussed here today. Again, my contact information is up there, and I'm looking forward to the Q&A session, but in the interim, I will pass it off to Charles.
Okay, can you see my screen here?
I'm seeing the desktop right now.
Okay, let's switch that. Okay, that should be better.
Yep, that works.
All right. Okay, so thanks thanks there, Brianne. And so my name is Charles Geddes, and I'm a research scientist in weed ecology and cropping systems based out of Lethbridge, Alberta. And today I'm just going to be giving an overview of a few of our research projects on which we are working. Okay, so Sean had previously introduced the glyphosate and oxinic resistant kochia surveys. And so I thought that I would show a little bit of data from Manitoba as well. So this is the same survey, but a different province. And so Manitoba was sampled in 2018, whereas Saskatchewan was sampled in 2019. And so we finished the screening for herbicide-resistant kochia in Manitoba. And basically the story with Manitoba was that glyphosate resistance was first discovered in Manitoba in 2013, and there were two populations shown by the asterisks here in this map. And those are found in glyphosate-resistant corn and soybean. And then we repeated the survey 5 years later in 2018, and we now reported glyphosate resistance increasing from 1% to 58% of the populations that were sampled in Manitoba just after 5 years. So glyphosate resistant kochia is quickly becoming a very problematic weed across the southern prairies. And in addition, we also found some dicamba resistance in 4 kochia populations as well. 2 with dicamba resistance alone, and then 2 with both dicamba and glyphosate resistance in Manitoba. And over here, this map is just another way of visualizing these data. Instead of reporting which populations were resistant, which were susceptible, here we're looking at the frequency of confirmation of glyphosate resistance within each rural municipality, showing that we generally have a greater frequency of confirmation of glyphosate resistance in the southern municipalities within Manitoba. One of our other projects is looking at oxinic herbicide resistance in kochia. So this is a rather new project that we just started this year. And as part of this project, we're looking at characterizing oxinic resistance. Is there variability among synthetic auxins? And also looking at determining the mechanism of resistance, and also looking at do we have PPO inhibitor resistance in kochia on the prairies? And I'm happy to report that it hasn't been found on the prairies to date, but we do have a surveillance program that's up and running because we do expect the group 14s or PPO inhibitors being sort of the next mode of action to which kochia will likely develop resistance just based on herbicide use patterns. So with this slide, I'm just showing 3 different populations right here in response to varying rates of either dicamba or fluroxypyr, just showing a visual example of how those populations vary in their resistance or susceptibility to synthetic auxin products. And this is showing the same thing over here, actually, but with more populations along the y-axis here. In response to dicamba or fluroxypyr, FW would be biomass fresh weight versus DW, which would be biomass dry weight. And you can just see that that these populations do vary quite widely in their resistance or susceptibility to these synthetic auxin products. And it's not necessarily consistent among these products either. Up here, just showing a map, again, the frequency of confirmation. In this case, it's fluroxypyr resistance in Alberta, showing that we tend to see greater frequency in this corridor between Lethbridge and Calgary. So some of our other work is looking at how to manage herbicide-resistant kochia in cropping systems as well. This is just an example of one of our studies looking at a spring wheat, canola, spring wheat, lentil crop rotation. And so this rotation is designed with a full herbicide layering package used throughout. And what we're interested here is, can we improve the ability of the crops to actually compete with kochia and reduce these kochia populations over time? So on the top, I have an example of wide row spacing with recommended seeding densities versus the bottom, which is narrower row spacing with double the recommended densities. And you can see visually the difference here. These photos were taken in 2020, but I have photos from the 2 years previous that show this exact same pattern, where improving the ability for these crops to compete with kochia can go a long way to depleting the kochia in the seed bank, going into a crop that doesn't have any post-emergence herbicide options like lentil. Another project is looking at diversity within crop rotations as well. So here again, showing a spring wheat canola, spring wheat lentil rotation. And we're also looking at other either 4-year or 2-year crop rotations. And looking at the diversity of crops within the rotation, but then also the diversity of the life cycles of those crops. So here, looking at the same rotation, but swapping out spring wheat for winter wheat. The idea being that winter wheat is already established in the spring, and you harvest winter wheat before kochia generally produces viable seed. So you're preventing seed from entering back into the soil seed bank here. And again, the idea being depleting the seed bank, going into something that doesn't have as many herbicide options like lentil. Here, comparing these two lentil crops with spring wheat before or winter wheat before. And this project is also looking at a perennial that would be cut 2 to 3 times per year. And we're going into the 3rd year of this rotational study currently. And actually this year in winter wheat and in the alfalfa meadow brome mix, you have to search very hard to even find a kochia plant within those plots. So this is good news. It means that despite the increase in resistance, it is still manageable with appropriate design of cropping systems. One of the other studies that I have the opportunity to lead is the prairie herbicide resistant weed surveys. And this survey basically covers the 3 prairie provinces, 800 fields over 4 years. And here just showing an example of wild oat and the group 1, group 2, and group 1 and 2 resistance in wild oat. And basically in this survey, what we do is a sampling similar to that was covered for the crop diagnostic lab, where we visit fields before harvest, collect seed samples, bring them back to the lab and screen them for resistance. The only difference is that this is a randomized stratified survey, so we can determine whether resistance is increasing or decreasing over time. And here we're showing an example of group 1 resistance in wild oat in the 3 prairie provinces. And so we just started the herbicide resistant weed survey, or the current round. Saskatchewan was sampled in 2019 and 2020, and we're working on screening those samples. And we're about 90% done the wild oat samples, and the preliminary data are showing that, at least in Saskatchewan, we're sitting around the mid 70s as far as percentages of field where wild oat, or percentages of fields where wild oat is collected that are confirmed to have group 1 resistance. So it is an issue that is increasing on the prairies. And this survey also covers a wide range of weed species. So I just have a quick list here of some of the top herbicide resistant weed species or biotypes in Saskatchewan. And of course, multiple herbicide resistant wild oat and kochia, so wild oat resistant to group 1, 2, and 8, as well as kochia 2, 4, and 9, are pretty big issues on the prairies. But then there's also other potential issues as well. So group 1 and 2 resistance in green foxtail, or group 2 resistance in a wide range of broadleaf weed species as well. And here, just the percentages, again, showing the percentage of fields from which that species was collected and tested, and those fields having confirmed group 2 resistance. So just 1/4 of the fields here for wild mustard, for example. One of our other projects just began this year, and it's looking at developing rapid in-season diagnostic tests for herbicide resistance. We saw the sampling and testing that's done by the Crop Diagnostic Lab, and basically this is providing more tools for testing where we could confirm resistance in season. And the idea being that after a farmer sprays the field, they suspect that they have herbicide resistance in a particular species, and they would be able to collect leaf tissue from that species, submit it to a diagnostic lab. The lab would extract DNA from the leaf tissue and look for either target site mutations or copy number variation, and in that way be able to confirm herbicide resistance in the season. And the idea is hopefully that this entire process from sampling in the field and getting a result back would take about 1 week. So the farmer would have information on some potential rescue treatments as well and what might work in that field. This is just showing a quick example looking at copy number variation of EPSPS in glyphosate resistant kochia. So basically, as there's greater copy number of EPSPS, it requires a greater dose of glyphosate to manage that kochia plant. One of our other projects is looking at weed seed suppressive soils. So we're looking at using a couple long-term experiments here at Lethbridge that were established 30 years ago. And in those experiments, they basically scalped out the A horizon of a field and deposited 36 different topsoils in a replicated design in those fields. And initially, this was used to look at how different soil properties impact crop productivity. But what we're doing is we're actually doing targeted burials of either wild oat or volunteer canola within these plots, and then leaving them for a certain amount of time and coming back and recovering those seeds. And the idea is that we would bury the seeds and come back to that same location, sample the soil in a core sample, and we use this elutriator that we built along with Artec. And basically what you do is you put the soil sample in these little strainers. This carousel turns through a water bath with high pressure sprayer nozzles spraying on the strainers. And what you're doing is you're washing out the soil from the sample and left with seeds that remain in the soil seed bank. And along with this, we're also looking at various chemical, biological, and physical soil properties, trying to determine what is it about a soil that helps manage weed seeds or promote decay of these weed seeds within the soil seed bank. Another project is looking at weed phenology, and more specifically, the phenology of weed seed production. So this project is looking at various common weed species across the prairies at 5 different locations. This is just an aerial view of Brienne's location in Lacombe. And what we're doing here is we're using 2 experiments that are overlaid here in this figure. The first one is where all of the species is planted all at the same time in the spring, and we have harvest dates progressing throughout the year. And what we're doing is each harvest date, we're quantifying the number of viable seeds on the plants. So we can look at when does the seed on the plant actually become viable. And then the other experiment is actually just a series of planting dates that are all harvested at the end of the year. And again, we're quantifying viable seeds, so we can look at how late does that weed species actually emerge and still produce viable seed before the end of the year. And the idea here, this is just an example for a couple site years of kochia. And the idea is that overlapping these 2 curves, we can identify sort of a target window where we can come in and manage kochia populations and mitigate the production of viable seed or seed entering the soil seed bank. And for kochia, these preliminary data suggest that it's somewhere around mid-August is that optimal management window. And the last study that I'll show here quickly is looking at developing an integrated weed management package for soybean production on the prairies. And we have various experiments looking at everything from variability among cultivars to row spacings, seeding densities, looking at fall rye cover cropping, crop sequencing, stubble residue management, and planting dates in various experiments. And this is just an example of one of the experiments looking at a slender versus a bushy type cultivar in narrow versus wide row spacing, in recommended versus double the or 1.5 times the recommended seeding density, with versus without a fall rye cover crop that's terminated at planting. And here we're just showing the plots later on in the season too. So the idea is basically determining what agronomic practices contribute to the ability for soybean to compete with weeds, and then also the soybean's ability to withstand competition or mitigate yield loss due to weeds as well. And with that, I'd just like to thank all of the funders of our research program and the co-investigators for all of these projects, as well as the technical staff and students that work tirelessly on these projects as well. So thanks for listening.
Awesome. Well, thanks, Sean, Brianne, and Charles for those presentations. And we will get into the questions. We do have a few coming in here. The first one here would be, I guess kind of general, anyone could answer this one if they feel like taking it on, but can herbicide resistance in problematic weeds be prevented by diversifying your herbicide applications and implementing herbicide rotation?
So this is Charles here. Maybe I'll at least start off with this. So there's been several studies that are looking at what we could call sort of integrated herbicide strategies, right? I think there's an important distinction here that is not the same as integrated weed management, because that entire strategy focuses on herbicides alone, right? So it's not a complete solution, but there are studies showing that, for example, mixing multiple herbicide modes of action or layering multiple herbicide modes of action can be effective at helping to mitigate selection pressure for herbicide resistance. But it's important to also consider whether resistant weeds are present in the field and know which herbicide modes of action are actually effective on those weeds. For example, if you have glyphosate-resistant kochia, just adding in a PPO inhibitor to your pre-seed burn down is not that effective. It will be effective in control, but it will also be selecting for resistance because you would only have one mode of action that is effective, right? So those integrated herbicide strategies can be effective at helping to mitigate, but it's also recommended that that other types of management, like cultural management or mechanical management, for example, are included.
Awesome. Thanks.
I'll just
ahead,
type in quickly. Yeah, sorry. That staging is also very important, and applying the right herbicide at the right stage of the weed's growth cycle is important, because as those weeds get too big, they become more tolerant, and then if they survive, then it can move on to the next generation. So if you're changing modes of action, it's important to have a look on that label and see what the proper staging is, because it can change quite a bit. I know I've looked at labels for kochia, and it'll go from 2 to 5 leaf to 15 centimeters. So if you have a product that needs kochia to be small and you spray it when it's too large, it will be lost. So yeah, I think that's just an important consideration as you are trying to switch modes of action as well.
Awesome. Thanks. This next question is for you, Sean. The AC Saltlander establishment challenges, can you speak more to them? Were seeds broadcast and incorporated, or just broadcast on top, or were the plants choked out by the kochia?
That is a good question. We went out and the kochia was already up. It was 3 to 5 leaf stage approximately. There's a couple sites where it was a touch bigger, but it was all around that same size. And we had broadcasted over the surface and we had raked it in. But I think it was a combination of it being dry and there wasn't enough moisture to kind of imbibe the seeds and have it germinate, as well as soon as the kochia was up, it was very thick. So I'm not quite sure if it did emerge and was choked out, or if it just was too dry and that rain came too late. So into the fall, I am hoping to mow back that kochia and try to reseed it to try to salvage those plots.
So this is Charles here, if I may add to that just briefly. So some of our rotation work, I showed a slide there with alfalfa and meadow brome. So it's not a sea saltlander, but what we noticed through the first 3 years of that study anyway, is that the first 2 years, kochia was actually very dense within those plots. And basically we tried to establish the alfalfa and meadow brome. When we seeded it, basically we had to do repeated cuttings of the kochia within those plots over the first 2 years, trying to prevent any seed from going back into the soil seed bank. And now in the 3rd year, there's very little kochia in those plots. So I think the real knowledge gap is how to effectively establish some of these crops in saline areas, right? And manage kochia over those first couple of years while the crop's establishing.
And one additional thing that I'd probably add to that is that there is some suggestion that some of these salt tolerant varieties of forages are a little slower to get established, and that is inherent in their salt tolerance. So there's some strong linkage there between the salt tolerance and their ability to get established, and so they take a longer period of time, maybe multiple seasons, to actually get a full establishment in place. So that could have a contributing element to that research that Sean's doing as well.
Awesome, thank you. I guess this could be like a two-parter here for Sean. For the straw side of things, first off, will burning excess straw after harvest help reduce or destroy weed seeds? And then second part, what type of straw was used in the kochia suppression, and will the type of straw make a difference on that?
Okay, I'm going to start with the second question first, since it's a little easier to answer. So we got out around late May, so there wasn't as much chaff around as I had liked. 4 of the sites had wheat chaff, either from a neighboring field or from inside the field itself. One of those sites actually had a bit of chaff from weeds as well mixed in. One site had canola, and at one site it had been tilled, so we actually pulled cattails from the margin and had used that. And we were aiming for 5 to 7 centimeter depth. I don't quite know how much that is as far as volumes go, as we were just kind of piling it on until I thought it would be good. Yeah, I think that answers that. As for burning, I might pitch this over to Brianne, since she has a little more experience with weed seed in chaff, but I would say it would be pretty effective. I'd be a little concerned about the environmental impacts, and I'm not quite sure if there's regulations around burning here, which might be a better call for the provincial folks.
Yeah, I can jump in on this, Sean. So in Australia, one of the first harvest weed seed control methods that they were using was dropping the straw and chaff into rows behind the combine and then burning them. It can be very effective, but you need to have a hot, slow burn to actually destroy the weed seed viability. So if you have a windy day and it takes off and just burns really quickly, it doesn't get as hot, and those weed seeds do not necessarily die. They were finding issues with some nutrient concentrations where they were having burns, concentrations and deficiencies, I believe. And really everyone has kind of dropped that method because there are easier, more effective methods that, although they take maybe a little bit more capital investment, they're doing a better job and they're less risky in terms of fire escapes and things like that. We haven't done research on the burning in Canada because of concerns around our environment at harvest time because of how dry it gets. In Australia, they will typically wait until they've had some rains. They will wait for just the right weather, the right wind direction, that type of thing. And we don't really have that same luxury of time as what they do there. So it can work, but concentrating is usually better because it gets hotter and you need it to be a hot, slow burn to have efficacy.
Awesome, thanks.
Yeah,
While we have, sorry, Clark,
I'll just add just a quick bit there. Yeah, just to kind of add on to what Brianne was saying. Unless that material is concentrated into a row, just burning the field itself is not really a good management strategy because one, it's too fast and your fire doesn't get hot enough. So just to kind of add to what Brianne was saying.
Brianne, we have another one here for you. Has the research of seed destroyer equipment on combines resulted in selection of very early maturing weeds?
So there has been some evidence for a shift to earlier maturing weeds in Australia. Primarily evidence in wild radish. They haven't seen it as much in ryegrass, is my understanding. But it's been in areas where this has been used for, over 10 years at this point. So it does take some time. Any management practice that you're using that's sort of your sole control factor will result in a selection like that. So if harvest weed seed control becomes the major focus of weed management, yes, you will see selection for earlier maturing weeds or earlier shattering weeds. So the idea is to really incorporate it in an integrated strategy where you still have, effective herbicides that can manage those maybe earlier maturing weeds. You have harvest weed seed control to manage what is herbicide resistant. You use your increased seeding rate and your good crop rotation to help manage those other biotypes that are maybe escaping some of those other strategies. So the more things that are included in your management strategy, the less likely you are to have those biotypes that are escaping your control. It's really kind of how we've gotten into the issue with resistance, is we've relied solely on herbicides or mostly on herbicides for our management. And so now that's escaping our primary management tactic, and now we have issues. So if it's the sole management strategy, yes, there will be some selection, but there are ways to manage that as well.
Awesome, thank you. Next question here. With the soil type effect on weed seed study, are you rolling or packing the soil profile after burying the weed seeds?
Okay, so basically, in this study we tried two methods. The first one that we tried was a mesh bag method, where we developed these mesh envelopes where we put the weed seeds in the envelope and then buried the envelopes in the soil. And we actually moved away from that because for this study we really needed to get the seed soil contact in there. So what we actually do is we have these plastic rings, and we take a core sample from the soil, and we place the ring down within that core sample and add 100 seeds, weed seeds within that ring, and then we replace the core sample back on top and basically pinch the edges to mitigate any potential preferential draining. So it's not necessarily rolled or anything like that. It's basically trying to take the core sample out, put the weed seeds in, and then place the core sample back on top.
Awesome, thank you. Next question here. How do we report a herbicide resistance found in a field? Is there a form we would need to fill out?
So I think there's a couple different aspects to this question, and basically the first part is making sure that the weed is actually herbicide resistant, right? So utilizing some of the diagnostic labs, like the crop diagnostic lab, to confirm that that population is actually resistant. And at this point, once you receive those results back, there's really no system that aggregates all of the resistance data or information across the prairies necessarily, other than the herbicide resistant weed surveys or a report that's coming out of the crop protection lab, for example. But that, I think that is sort of a gap, and one of the areas that I'd like to focus on is developing a system where we can actually have reported confirmations of herbicide resistance, and where these biotypes occur across the prairies, that would aggregate information from the prairie surveys and also grower submitted samples.
Awesome, thank you. That is actually all the time we have for questions, so we apologize if we weren't able to get to your question. I want to thank Sean, Charles, and Brianne for their presentations again, and thank you for taking the time to join us here today. Our last speaker of the day here, it's going to be Clark Brenzel coming back for round 2. He's going to be giving a presentation on herbicide injury. And Clark, if you're ready, we'll let you take it away.
Okay, you can hear me, you can see me, everything's good?
Yep, looks good.
Okie doke, away we go then. So what we had at the ICDC location in Outlook, we had a series of plots go out looking at various injury scenarios. Some of these worked and some of them didn't. Essentially, I'm showing the ones that worked, and kind of looking at some of the injury symptoms that we're seeing at those plots. First, I'll kind of do a little bit of a review of what Shannon had kind of gone over with her herbicide resistance, because there's a lot of parallels in some of the discussions about resistance and injury, in that we use a lot of the same terminology as far as how to refer to the herbicides. So to basic level, what's a herbicide? It's a product that's used to control weeds. Generally, it's something that's applied to the weeds or to the soil in order to interfere with the life cycle of the weed, essentially. That isn't a physical or manual type of removal. So it can be a chemical compound or it can be an organism in the case of a bioherbicide. And generally what they do is they interfere with a critical pathway in the plant. That means that the plant will fail to continue its life cycle, so it'll end up either being suppressed and stopping at a certain stage for the rest of the growing season, or the plant will outright die and end up reintegrating into the soil system. So another little piece of terminology is that a selective herbicide is one that will kill a weed within a like desirable plant. So where you've got group 1s that control only grasses, you're looking at controlling a grass within another grass, or you could be looking at controlling broadleaves within a grass crop. You could be looking at controlling grasses within a broadleaf crop. So that's the kind of concept of selectivity that we're talking about. Generally, when we look at herbicides, they'll attack a critical pathway in the plant. They're not going to attack a minor pathway in the plant because the odds of success of really sort of hitting that plant where it lives is pretty minimal. So generally it goes after a critical pathway. And a lot of those critical pathways have already been explored, and that's why there's challenges now with finding new herbicide groups that will actually find different mechanisms to control weeds. And so that's why we really have to focus on looking after the herbicide groups that we have from the resistance perspective. So a group is essentially a series of related herbicides that generally attack the same site within a plant. And if that plant modifies that site in a resistance kind of scenario, then it affects all the herbicides within that same group. And so we can identify those groups in a couple of different ways. Australia used to use a lettering system that was different from the Herbicide Resistance Action Committee, or HRAC. They've recently shifted towards adoption of a numbering system that is very similar to the one adopted by the Weed Science Society of America. And so those are right now we have the Weed Science Society of America system, and we have the Herbicide Resistance Action Committee lettering system that are available for describing herbicides. So that kind of gives a little bit of a background. The graphic that Shannon had showed earlier about how our herbicides work, we talk about enzyme systems. Again, two parts go in and a new part comes off. The flip side of that is it could be a detoxifying system where that larger piece goes in, gets cleaved in half and moves away. And Shannon had kind of looked at that a little bit as well. And then where the herbicide inserts in there, and we get a backlog of that system, or we get our critical component not being produced, that would be more of our systemic type products or our growth inhibiting type products. And those are all listed on the right-hand side there. You note the asterisks on group 10 there, or the glufosinate. The other system that Shannon talked about is where you get something backing up within that process, and that ends up being toxic to membranes in the cells of the plant. And so those are the products on the left-hand side that we're talking about there. And again, you'll see group 10 shows up on that side. And essentially what that means is that glufosinate can act in both ways, in the sense that what you're doing is you're inhibiting the production of things that are related to glutamine. So it's interfering with the glutamine synthesis process. And as a result of that, you're getting ammonia backed up within the cell, and that's toxic to the membranes. And so you're getting that contact type membrane disruption type of herbicide activity. Then we also have some herbicides that are multi-target site, or they're non-target site type herbicides. Group 3s bind directly to what's called tubulin. And tubulin is a compound that shows up during cell division, and it's what pulls the chromosomes apart when cells divide. And so as a result, cells don't divide properly when that herbicide is present, and so you end up with sort of a building up of material that really isn't really well defined cells. Group 4s are auxin mimics, and so essentially they're reacting in the plant the same way that the natural hormone would react in the plant. The difference is that the plant can't break that down, and so it's a cascade of effects, and so the plant succumbs to its own hormone, essentially. The other thing to keep in mind, especially when we're talking about injury in plants, is we have to kind of keep in mind as to how that herbicide is moving within the plant. And so there's two different methods. There's the apoplastic movement, and that's essentially passive movement within the xylem of the plant. So it's generally all upward. So it's moving from the roots to the leaves, moving with that water. And when you have herbicides moving that way, they're not going to move from one leaf to the next leaf. What they'll do is they'll either move from the roots upward in the plant and collect at the periphery of the leaves, or we'll look at the contact system later. If we're looking at a symplastic movement, that's something that's moving largely in the phloem of the plant, or and to move in the phloem of the plant, you have to actually enter the cell first and then move in between cells through these little gaps called plasmodesmata, and then into the phloem where it's actively transported within the plant. You can get both of these happening at the same time. Generally with apoplastic movement, you don't have to have the material enter the cell. It can actually move in the intercellular areas up into those areas. So if you do a post-emergent type of application with apoplastic movement, that really doesn't go very far, because again, you don't get movement out of that leaf and back towards the core of the plant. But when you have symplastic movement and it's moving in the phloem, it can actively move towards the growing points, and essentially you end up with a concentration of that material in the new growth of the plant. And so that's why you'll see certain symptoms within certain types of herbicides. The other terminology that we get is that if something's moving up in the plant, it's acropedal, and if something's moving down on the plant, it's basipedal. So you'll see me refer to apoplastic, symplastic, and apoplastic and acropedal both kind of synonymously, and symplastic not necessarily is synonymous with basipedal. So let's look at group 1 and just a little bit of background on group 1 herbicides. They inhibit the acetyl-coenzyme A carboxylase pathway. And essentially this is responsible for the production of fatty acids within the plant that essentially create membranes and waxy substance like the cuticle on plant leaf surfaces. These things move actively in the symplast. So again, they move from the leaves to the growing points, and that's where all the activity happens. The treatment that we had in the diagnostic school plots this year was that there was quizalofop, which goes by the proprietary name of Assure, applied at about 10% of the rate, so 30 milliliters per acre, and then also added an adjuvant like Shermix at half a liter per 100 liters of carrier water. And essentially the idea here is to simulate drift. These were applied to corn plants. And so we get a variety of different injury symptoms showing up. The picture on the left is something that we see in not only corn, but we see it in wheat and barley and other cereal or grass type plants. Essentially is that yellow band shows up on the leaves. You find that typically more with selective type group 1 products, again, products that control grasses within other grasses. And you see that showing up in situations where the growing conditions may not be as optimal as they might be. So you've got cold conditions, you've got maybe saturated roots, sometimes under hot conditions. Generally, it's the cold and saturated roots where you see it more often. And essentially where that band shows up is where that plant was unable to break down that herbicide for a short period of time. Once the weather conditions turn around, then the plant can sort of regroup, get all that stuff under control, and then you've got that white or yellow band that progresses up in the canopy a little bit. What you really kind of can see in a lot of grass plants is that where that leaf was in its development phase is where that band occurs. So on older leaves, it's closer to the base of the leaf, and on newer leaves, it's closer to the tip. And that's just essentially what was being produced at the time when the herbicide was active. And so the other symptom that we can see, particularly in corn, is some of the loss of that membrane integrity. And so maybe if we look at, where's our pointer here? So if we look at some of this area down here, again, similar to the band, this is the area that was in production at the time when the herbicide was active. And so you ended up with cells not being produced that were very viable cells, and so you end up with interruption of that, the production of that tissue in that area. So it almost looks like you've had a hail on that and it's gotten thrashed a little bit. We're also finding that the plants are not as well developed in that area, and so they're constricted and prevent the new leaves from emerging from the old leaves. And so we end up with things getting stuck up here, and that can result in the leaves starting to arc out this way. And so you get into the symptom called a buggy whip type of symptom as well. And so you'll see that not only group 1 injury in corn, but you'll also see it in some of the group 15 type injuries as well. And largely that's because group 15 interferes with a very similar pathway that interferes with the production of fatty acids that also result in production of membranes and cuticular wax. So those are the kinds of symptoms that you see. These little ripples here on the leaves, generally what those are is that the plant was actually growing very rapidly, and you'll get these ripples showing up when plants grow very quickly. Sometimes if you end up with some tissues growing faster than other tissues in some spots, you'll end up with these ripples showing up on the plant as well. But you can get these ripples on plants that have no herbicide injury. So group 4, again, we talked about those earlier. Those are the things that mimic natural plant hormones. And essentially what they do is they result in unregulated elongation of the plant. So auxins are responsible for cell elongation within the plant, and it makes it so that the plant looks as though the internode areas are longer than they should be. So you end up with leggy looking plants or plants that the ratio of the width of the stem to the length of the stem is disproportionate on the length side of things. The other thing that can happen, particularly at the lower stem regions of the plant, is you get undifferentiated callus cells being produced, or callus tissue that really doesn't know what it wants to be when it grows up, but in the meantime it's bulking up in between those areas. And with broadleaf plants, what's happening is that you're getting those cells being produced, and they end up crushing the xylem or water transport vessels within the plant. And essentially what they do is they choke the top of the plant off from the bottom of the plant. The other thing that you'll see is you'll get the leaf cupping, you'll get stem twisting going on. So here's an arc in this soybean leaf where you get the stem twisting in an unnatural fashion. You've got some of the looping going on in that top soybean plant up here, where you've got some arcing going on. And there's also some cupping of some of these leaves here going on as well. Not quite as strong as what we would expect it to be with, say, the phenoxys. In this case, the treatment that was applied was clopyralid at 85 milliliters per acre, and that's the 360 gram per liter one. So that would be the lowest use rate that Lontrell typically gets applied at. And this was applied to soybean. And I talked about that crushing mechanism. Cereals are tolerant to that crushing function, not so much because they're necessarily immune to the herbicide or the group 4 herbicides, but because their physiological arrangement is a little bit different and they tolerate that crushing a little bit better. They still can exhibit symptoms. And this is a treatment of dicamba at 73 grams acid equivalent per acre. And that's about 1.3 times the label rate. And we did that just to try and stimulate some injury symptoms. We're within the label staging here. This is at the 4 leaf stage. Generally with dicamba, between the 3 and 5 leaf stage is the recommended timing. Usually if you get injury like this, it will be if you've gone outside of that normal leaf stage, but we generated it based on a little bit higher than normal rate being applied. Generally what you find with the symptomology on cereals is that it essentially looks like the plants are suffering a bit from drought. But keep in mind that this site here is an irrigated site, so there's no shortage of water in this system. But on the barley plants that have been treated, you've got kind of a silvery gray color change on the foliage, so it's a lighter colored foliage, can kind of trend into the blue spectrum a little bit. You end up with shorter leaves. In order, again, everything that's being done by the plant as a result of this is to try and preserve moisture loss, because it thinks it's a drought, because there's a restriction of that water moving up from the roots into the top, not a total complete cut off of the water, but just it's slowing, so it's experiencing droughtish type symptoms. So shorter leaves to reduce evapotranspiration, fewer tillers, because again, it doesn't have that free-flowing water in order to allow those things to develop properly. Sometimes after group 4 applications on cereals, you'll find that they flop, or rather than standing up erect, what'll happen is they'll lay flatter to the ground. And if we went further into the season, particularly if we were outside of those normal staging timelines, the heads may have aborted kernels on them. The heads may get caught in the flag leaf as they emerge, and that's when you get the head and on kinking that happens. It's very stereotypical of group 4 damage. We also looked at the impact of MCPA on corn plants. And so we've got 4 different treatments here. So we've got an untreated control, or glyphosate by itself. We've got glyphosate in a tank mix with MCPA amine, either at the 4-leaf or the 6-leaf stage, and MCPA by itself at the 8-leaf stage. I don't know that I would get too hung up on the particular size of the relative size of the plants, other than the glyphosate and the MCPA amine at the 4-leaf stage, and that the stem is somewhat narrower. That could be an impact of the phenoxy on that plant. But the thing to really kind of notice is around these root areas here, where if we look at the untreated plant, we've got lots of secondary root development off of those primary roots. Whereas with particularly with this 6-leaf application, where you're getting right up to the boundary of the timing that you should be putting MCPA on at, you're finding that we're getting fewer of those lateral roots and those secondary roots being produced onto those major roots. And so you're seeing some of the impact of the phenoxies that way. The other thing that you can see is on these 4-leaf stage ones, the prop roots are not really developing the way that they should on corn. So that's another symptom that is pretty indicative of that. We're seeing worse injury in this situation with the glyphosate mix, and essentially what that could be is that the additional adjuvant in the glyphosate is actually making the MCPA amine act more like an ester, and so we're getting quicker uptake of that MCPA into the plant, and so a more rapid onset of injury symptoms. The other thing that isn't really visible in the picture here is that phenoxy injury to corn plants also results in brittleness, and we'll also see that in exposure of broadleaf plants to phenoxies as well, where the plant gets very, very brittle, and so there's not very much flexibility in that plant. So you can snap that tissue very easily, and that with corn here in particular makes it more susceptible to lodging, being a really tall and brittle stem. Group 9, essentially Group 9 is glyphosate. It's the only compound in the group. There was some discovery work done way back in the day when glyphosate was first developed to look at other compounds. And really glyphosate was the only one that kind of met the bar as far as being relatively good activity and also relatively good user safety and health profiles and things like that. Glyphosate inhibits EPSPS, and that's responsible for the production of amino acids. The amino acids that are being suppressed are tryptophan, tyrosine, and phenylalanine. You'll get, because you're affecting branch chain amino acid production, very similar to what you get with group 2 herbicides. The symptomology with glyphosate injury is also very similar to what you would get with group 2 herbicides. So generally, you get a cessation of growth very quickly. You get immature leaves in the growing points being affected first within about 5 to 7 days, and then essentially you're inhibiting the production of something that's essential for growth. And so where those new leaf tissues are really in high demand of that material, they'll go down first, and where the older leaves have already laid down that material initially before the treatment, they're not going to require it until they have to go and repair the tissues later on. So the impacts that you'll see are yellowing and new growth. And again, that's because it's the first stuff to experience deficiency of that amino acid. Stunting and miniaturization of the newer growth, loss of apical dominance. So that means you get eruption of lateral buds all up and down the stem, and compression of the newest internodes. Glyphosate is moved actively through the plant in the symplast or the phloem, and so it'll move directly to the growing points. The treatment that we had in this situation was on Liberty Link canola, and we put a rate of 18 grams acid equivalent per acre, or 1/10 of the half liter rate, or half liter equivalent rate, which is a term that I don't really subscribe to, but I'll use it in this situation just to kind of give some perspective. And this was applied at the 4-leaf stage. So essentially, what we're seeing is that the primary symptom is in the new growth, where we're getting some growth distortion, and in some cases you can get what's referred to as auxinic effects, where you can get cupping and curling of the leaves. And so if we look a little bit closer at some of those leaves in the main growing point, we're getting cupping happening in there. And we won't necessarily get color change to the new growth at sublethal rates, but you'll get some of that growth distortion activity taking place in there as well. Note, just to kind of go back a little bit, again, don't focus on the purple color change, because purple is just the plant's cry for help. It could be many, many different things that are causing the problem, herbicide injury being one of them. So again, don't really focus on purple being the problem. It's these impacts down here in the main growing points that are the things that you want to be looking for. These plants aren't big enough to really be showing any branching changes, but we'll get into the next slide here and maybe we'll see something. Once we start getting recovery from a glyphosate exposure, they're still not really going to grow as effectively as they might. You're still going to get, again, miniaturization up here in the main growing points. You're going to get stunting of the stem. Again, we're getting some of those auxinic effects with some of these little curvatures in the stem. The little bit of back and forth curvature in this stem is normal. If you start getting to like arcs of this degree, or some of the arcs that are happening down here in the base, that's abnormal for a canola plant and its growth. And we're also getting some of these, some growth coming from these branches a little bit as well. But mostly what we're getting here is miniaturization, kind of not really getting good flower production. We're getting some curvature of the stem, and that's on recovery from glyphosate exposure. Group 10, again, glufosinate is the only product in this class that's really relevant to Western Canada. And essentially it's inhibiting the production of glutamine within the plant. And that does two things. It prevents the production of essential amino acids, as we talked about earlier, and also backs up ammonia within the cell, and that results in cell rupture and rapid necrosis of the plant. Generally, because it's an amino acid inhibitor, normally it would be moved around the plant via the symplast, but because of the rapid cell rupture, that process is self-limiting. And essentially what it means is that that stuff gets stuck in the leaf, that cell rupture happens before it's able to get that material much moved out of that leaf and throughout the plant. It's not very strongly bound to soil, but it breaks down very quickly in soil. The treatment we've got here is a quarter of the 1.35 liter per acre rate of the 150 gram per liter formulations. And that's applied to the unifoliate and trifoliate leaves of soybean and the rosette of Roundup Ready canola in this situation. So for our, at our unifoliate, you're really not seeing a whole lot in this case. It looks like the plant has pretty much recovered from that early application, and that's not inconsistent with something that's quasi-tolerant to a cell membrane disrupting type herbicide. And it's because of the low rates that we're using of the glufosinate in this case, that the plant has actually recovered quite nicely. There's a couple of other things going on. It's a little bit paler than normal, and that's kind of the glutamine inhibition that's going on. And there's a little bit of crinkling on the leaf tips down here as well. But outside of that, this plant has largely recovered from that initial treatment. And that was about 10 days out. If we look at a plant that was treated a bit later in its life cycle, now you're getting more distinctive symptomology that you would expect to see. After 5 days, we're seeing quite a bit of necrosis here from cell rupture. We're getting some of that crinkling that we saw from the earlier application here at 5 days out. And so the plant looks like it's pretty much struggling here. But by about 12 days after, you're getting a few new leaves being produced, and they look, again, relatively normal. They're a little on the pale side, but generally they're largely normal. And I would expect these plants to pretty much recover after this application and continue on nicely. In canola, essentially what we're seeing in this case is we've got the untreated on the left and the treated on the right. And so we've got some spotty necrosis taking place, not enough material in the application to really cause complete necrosis of the plant. But what we're seeing is we've got some spots on this big leaf down here that was present at the time of application. We've also had some new leaves be produced after the fact. There's a little bit of mottling on some of those leaves because of the glutamine inhibition. We're seeing some actually bleaching symptoms on the outside of the leaf, and what we're seeing is some acropetal movement of that material from the center of the leaf to the outward edge of the leaf, where it's concentrating and providing more injury symptomology. And we're seeing that pretty consistently through a number of leaves in this plot. So it can kind of mimic the injury of some other things that are some of the bleaching compounds. Some of the herbicide injury look-alike, and this is pretty relevant when we were just talking about glufosinate injury. Adjuvant burn can cause a loss of cell membrane integrity if it's fairly high. Sometimes we get adjuvants acting a little bit like, and water droplets acting a little bit like magnifying glasses. And in some cases, you can get some bleaching of leaves as well as a result of adjuvant burn. Some other things that you might see in the field are things like genetic albinism, where you've got cereal leaves where you've essentially got single veins running up and down the plant that are either white and alternating white or green. That's a good indicator of albinism. So essentially down in the growing point, there's been a genetic mutation at some point, and it's lost its ability to produce pigments. And similar in these canola leaves here, you can get various degrees of paper white tissue, and in this case, it's only on one half of the leaf. So at some point down here, the main growing point got impacted, and any tissues that kind of come off of that point where that growing point was injured end up staying either white, or they lose the large majority of their pigments. Drought is very similar to the impact that we see on cereals for group 4 injury. So again, you can see the shorter leaves, and you'll end up eventually getting leaves sloughing off the bottom here in order to continue the growth of the top part of the plant. And the goal of this plant is to produce seed at the end of the day, so these leaves really don't matter to it. So it'll sacrifice these leaves in order to continue movement upward in the stand. And that's very similar to what we're seeing if we get a very severe injury from a group 4 herbicide. Also, what we'll see come into our lab occasionally as concern about herbicide injury is things that are consistent with nitrogen deficiency. So we'll get older leaves yellowing off, new leaves being perfectly green and perfectly formed, but the older leaves are yellowing and starting to drop off and being necrotic. Same thing with cereal plant. New leaf tissue is green, might be pale green, but it's green still. And some of these older leaves are starting to start to die back. And generally that happens from the tip, moving back to the base, and eventually they dry up and drop off as well. The other thing that we see with group 4s is the kinked heads, and there are some varieties that there is a small percentage of those plants that will do that naturally. You still have the same complement of kernels within those heads, but for whatever reason, when they're growing very quickly, they get caught in the sheath of the flag leaf, and they end up with kinked heads. And you're looking at maybe, if you want to compare injury to the natural kinking, you're probably looking at, with natural, about 1% of heads would be affected. And with herbicide injury, you're looking at maybe a quarter or more of the heads would be showing symptomology. And with sticking with group 4s, something that happens out there, sulfur deficiency can resemble, again, we talked about with the purple coloring. So with sulfur deficiency, we also get purple coloring of the leaves, but we get an upward cupping of the leaf in this case. The stem leaves also will kind of cup upwards with sulfur deficiency, and that can be confused with group 4 injury, sometimes with glyphosate injury as well. Another thing that we see from time to time, especially when it's wet, probably won't be seeing that this year very much, in lentils is something called wet feet edema, and that can kind of look a little bit like a group or a membrane disruptor herbicide drift, where you get a bunch of little spots on the stem. So you get little random spots occurring. Essentially what's going on here is that you get a very, very high influx of water into the plant, and that causes the cells to burst, the little individual cells to burst, and you end up with these little white spots on leaves. You can also get it in cereals, and in cereals it's generally referred to as physiological leaf spot. And that's all I had for today. If you have any questions, I'd be happy to entertain them.
Awesome, thanks, Clark. Yeah, we do have a few questions coming in here. First one for you, could you give an example of a bioherbicide?
Some of the work that's being done at Ag Canada, they're looking at organisms that can be applied to things like scentless chamomile. There's some work that's been done on Downy Brome by Suboyechko. And the one that's actually hit the market is there was a product that was developed by Ag Canada out of Saskatoon for application to initially thistles. And so essentially, if you look around the countryside and you see thistles with white tops, that's the same organism of the bioherbicide that, and essentially what they went out was at that time of the year, collected that organism, and essentially what you're doing is you're shifting that timeline and you're moving it towards earlier in the season. And so that's the one that is sold into domestic markets. I forget what it's called now. It's gone under several different names. Originally, Scott's had it for a little while as a bioherbicide for dandelion control. It's the organism's name is Phoma macrostoma, and some of the manufacturers actually use that Phoma as a brand identifier as well. So that's one that's actually made it to the marketplace. The other one, there was an agent that was being investigated several decades ago now on roundleaf mallow, and actually worked quite well. The real challenge is that bioherbicides don't work very well under dry conditions like what we have now. So if you've got high humidity and lots of rainfall conditions, they work reasonably well. But under fairly arid conditions, they tend to fail more often than succeed, unfortunately.
Awesome, thanks. So this one's kind of a two-parter.
Okay.
About for desiccants. Will the dry, dusty conditions create any issues with producers using desiccants this fall? And the second part of that is, when it's this dry, are desiccants really even needed?
Well, and that's the real crux of it, is that when it is as dry as it is now, I would suspect that a true desiccant really isn't needed. And when I have to qualify true desiccant from some of the misinterpretation of the use of desiccant in a lot of cases. Oftentimes we'll hear glyphosate being used as a desiccant, and essentially it's not registered for use as a desiccant. It's registered for use as perennial pre-harvest weed control. It essentially, a desiccant will dry the plant down within about 5 to 7 days. And essentially a desiccant is there to facilitate the timing of harvest. The case this year is that we're finding things are coming mature very, very quickly. And I suspect that the need for desiccants would be fairly low this year, like a true desiccant. It doesn't mean that there isn't opportunity this year to apply glyphosate to Canada thistle for control of that. But because things are coming in fairly early this year, may mean that there's more opportunity after harvest to do post-harvest glyphosate applications to thistles as well, and kind of avoid some of those market issues that are kind of emerging with glyphosate application prior to harvest.
All right, thank you.
First part of the question, I should answer that.
Okay.
Generally, if we look at things like dicot, they are they're very susceptible to being bound up by dust and by clay particles, so that can be an issue. I don't think that something like heat is as strongly bound by soil particles, and so it may not be as strongly affected by dust as something like a diquat type herbicide would be. Glyphosate activity can be very much impacted by dust on leaves.
Awesome, thanks. This one deals with Group 2 carryover.
Okay.
What are the typical symptoms of carryover damage, in particular flucarbazone? Does it affect the growing point and or stem?
It's very difficult to tell one Group 2 from the other as far as symptomology goes, and it can vary from crop to crop to crop. But in large general terms, essentially what you would get with Group 2 injury is miniaturization of new growth, loss of apical dominance. And so what that means is that you're getting all the lateral branches all erupting at the same time. And so if you look at a plant, to pull a single plant out, and if you've got equally sized branches, like right from the tip of the plant all the way down to the base of the plant, that's a good indicator that you've had loss of apical dominance. And that's not from having like a bunny or a deer come along and nip the top of the plant off, because naturally what will happen with a plant is if it loses that main growing point, generally the next one down will take over and start to dominate the other growing points below it. In this case, you're losing all apical dominance, and so all buds are germinating or erupting all at the same time. And from that perspective, you're probably even going to get multiple branches erupting from the same spot, because that main branch isn't now suppressing the secondary branch that's primordia that's sitting waiting to erupt down there in the base as well. So you end up with a proliferation of branches, potentially multiple branches from the same leaf axil. You end up with a constriction of the internodal distances. So what'll happen is your plant is going along, and then all of a sudden, maybe your internodes are a couple of inches apart. All of a sudden, now they're a half an inch apart, and so they'll get progressively shorter even as time goes on as well. And generally, the reason for that, where you may get normal growth, and all of a sudden it gets affected, is that you get a rainfall, and what that does is that liberates all that herbicide that's sitting with attached to the soil particles. You get a rain, it liberates all that herbicide from those soil particles, makes it available for the plant to take up, and all of a sudden you get a major exposure of that susceptible plant to that herbicide that it doesn't tolerate. And you end up with the impact after the plant has grown to a particular size. Does that make sense?
Yep, perfect, thank you. Next question here. This person hasn't heard of adjuvant burn before. Are there weather conditions that enhance adjuvant burn, or things you can do to prevent it?
Generally, it happens under very lush growing conditions. So you've got ample moisture. You'll have very thin cuticle layers because you've got ample moisture. The plant has no need to lay on heavy layers of wax in order to preserve moisture. And so you'll have very thin cuticle layers, and when you apply like an oil surfactant blend or a solvent type herbicide like Merge or Turbocharge or things like that onto those leaves, and what they do is they essentially denature the wax on the surface of the leaf, and then that results in the tissue underneath getting sunburnt, is essentially what happens.
Perfect, thanks. Have you found any resistant Russian thistle in the province, or I guess have you had any come through the lab either?
I think during some of the survey work, I I know in Montana they've found glyphosate-resistant Russian thistle. I don't know if we found any in Saskatchewan yet. There may be some that have been found in southern Alberta. I'm not sure if Charles is still on, if he can answer that question.
I think he had to take off
Yeah,
earlier
looks like he
there.
left. Yeah. So, it's possible to occur. Let's put it that way. That you can get glyphosate-resistant Russian thistle. It's very closely related to kochia generally. They're both in the same family as things like lamb's quarters, so they all have similar genetics to some degree.
Okay. And this will be the last question here. So if you
Okay.
do if you do go in with glyphosate after harvest, you need to wait a certain time to allow weeds to recover. This one, their situation here is looking at quackgrass and wheat.
Okay. Yeah, generally what we suggest is about 4 to 6 weeks of regrowth in order to get enough new green tissues in order to take up the glyphosate and have it be redistributed through the rest of the plant. Quackgrass might be a bit of a tough nut this year again, and under the situations where you've got dry conditions, because I know my hay crops don't look all that great right now, and I suspect that the quackgrass is probably going to be suffering a similar fate. So you may not get as good a control this year as you might in a normal year, unless you're in the southeast of the province and they seem to have ample moisture. Yeah, 4 to 6 weeks. Keep in mind though that even with that time frame, you still have to put on more than the 360 grams active or acid equivalent per acre that you would with a pre-harvest. You would have to put on about 2.5 to 3 times as much in order to get the same amount going into the plant, like into the root zone, because you still have less leaf tissue after the fact than you would before harvest.
Perfect. Well, thanks. Thanks again, Clark, and appreciate you taking the time to be here with us today.
Not a problem. Appreciate the opportunity.
So that wraps up day 3 here, Crop Diagnostics School. We'd like to thank you all for taking the time to join us for the event. Again, if you entered your CCA number, we're going to pass that along so you get credits for it, and you had to be here live to get the credits. We're going to be sending out the webinar recordings again this end of this week, so you can rewatch any of the ones you missed, or if you wanted to catch something again. If anybody has any questions regarding these topics or anything else related to farming, you can contact your local SaskAg Regional Office or the Ag Knowledge Center at 1-866-457-2377, and we'll be sure to put you in contact with a specialist to help answer your question. Join us again tomorrow, our last session on irrigation. If you haven't registered yet, you can check out prairiecca.ca for more details. Have a great day.